Camille, is there anything that you want to address specifically before? Okay, okay, so, on Thursday, your attendance question is like, what are you guys still struggling with? And so I went through them and I made this. So, so somebody was like, do we need to know junctions like plasmodesmata and all these other things? No, you do not. Not for me. So don't worry about that. Okay, what is the functional difference between DNA polymerase and DNA primase? All right, so this is getting into the enzymes involved in DNA replication. You guys ready to get confused? Here we go. DNA polymerase is the enzyme that actually incorporates new nucleotides while creating that, doing that replication fork, semi-conservative stuff, right? This is the actual hard labor. This is where we actually get a new DNA strand. Here's where the confusing part comes in. DNA primase, its job is to tell the, to make an RNA primer. So when this DNA starts to open up, it needs a little primer so that the DNA polymerase can hook on. Here's where you're going to get confused. DNA primase is an RNA polymerase because it's making RNA. Okay, who's confused? Right, there has to be somewhere for the DNA polymerase to attach to. That spot is an RNA primer. The DNA polymerase equals RNA polymerase, because it has to make that RNA primer.
Can you just say it one more time?
Yep. I can even write it on the board. DNA polymerase incorporates new nucleotides. That's one D. Let me just rewrite that. New nucleotides to make more DNA. Okay. Our DNA primase is actually, for realsies, an RNA polymerase. And its job is to create an RNA primer. So DNA polymerase can begin replication.
So if that was a test question, then we could just say DNA primase comes before DNA.
Correct.
And that would be sufficient, or?
Yes.
Okay.
So if we went through this in order, so we have our DNA strand, right? What's the first step we got to do? Right. Well, first of all, it's like this, right? So we gotta untwist it, right? And so that enzyme that untwists it, That's the one with a really long, confusing name that nobody can actually pronounce. Yeah, that guy. Topoisomerase or something like that. Okay, it's a really long one. Okay, so that's untwisted. Now we got. Now what do we got to do? Unzip it. And what's the enzyme that unzips your genes? Yeah, helicase. Right? Yep. Okay, so now we've got our leading strand and our lagging strand, right? Because they're anti-parallel, we can only replicate in one direction, and so then the lagging strand is screwed. Okay, so then what do we got to do? We got to say, here's where we're going to start. Here's our primase. Okay, here, make our RNA primer. Then DNA polymerase comes on, hooks on to where we have that primer, and copies and copies and copies and copies. And then we have to, on our lagging strand, we have to add that a bajillion times, right? Because we got those Okazaki fragments. Okay. So now we got a bunch of Okazaki fragments. What are we going to do about that? That's a problem. What enzyme is going to solve that problem? Ligase. Yep, ligase. It's going to come through and connect all of our Okazaki fragments. Okay. All right, very, very good. All right. When tRNA is being used, where does it get the amino acid from? Well, we kind of talked about this today. So we have here's our ribosome, here's our mRNA strand, our tRNAs are flying in. They've got our amino acids attached to them. They just get them from the cytoplasm. Amino acids are just hanging out in cell juice. And they just like get them because they like bump into them, right? Because just like random molecular movements. Yeah.
So the peptide chain, does that just go when the tRNA leaves?
Yeah, so here, this is,
back out into the cytoplasm?
so this is our new proteins chain that's being built, right? This is the whole ribosome's jam making proteins. We got our TNA comes in, clinks on, and then it leaves, it just floats it back out into the cytoplasm, bumps into the amino acid again, and then it bumps into the ribosome again, and then there we go. Okay, so the tRNAs are constantly cycling through. Okay on this? Oh, introns. What the WTF, man? Are we okay with this now? Everybody understand introns, extrons? Okay. Introns, we don't care about them. It's a complicated acronym. Whatever. Okay. All right. This is a good one. Does the length of the fatty acid tails of the phospholipid affect the membrane? It does in two ways. So way number one, remember we talked about, so the phospholipid bilayer creates a plasma membrane, right? It's the interface between the outside of the cell and the inside of the cell. Creates all these chemical gradients that we love. The factors that affect diffusion, we did a little experiment with the food coloring. What things did we find out? We tested temperature. We tested solute density or concentration, and we tested molecular weight. So one of the things that we did not test was the thickness of the semi-permeable plasma membrane. If you have a thicker thing, what do you think is going to happen to diffusion? It's going to take longer, right? So longer fatty acid tails is going to make diffusion take longer. The other thing that it does, Remember, the other thing about the plasma membranes is it's nice and fluid. If you have a long chain, it's going to make that plasma membrane more rigid. Going to decrease fluidity. Okay, cool. And then, of course, these nice little peaks in these fatty acid chains, those that unsaturated fat is going to increase fluidity. What are some other things that we remember that affect the fluidity of the plasma membrane? Cholesterol, temperature. So if I said something like, all right, so if I was going to analyze the plasma membrane components of an Arctic fox versus a jackrabbit that lives in the Sonoran Desert, what would we expect? What would we expect to see any differences between an organism that lives in a really cold place versus a really hot place? What would some of those differences be?
Like cell structure wise, or just?
Just the plasma membrane itself. What would we expect to see the differences in the plasma membrane of something that lives in a cold place versus something that lives in a hot place?
The fox would probably have more cholesterol.
It would probably have more cholesterol, right? Because it needs
More fat layers.
More-
the insulators, so.
So that's like thinking, you're thinking about the organism itself. I'm going to zoom way into the particular, just this phospholipid bilayer.
Well, that makes it
So-
fish, like in the fox.
And then the fox, yeah, so it would make it more rigid. So so something that lives in a cold place want a rigid plasma membrane? No, it would want a more fluid. So it would have, so it wants insulation on its body in the form of fat cells, but in its phospholipid bilayer, it wants shorty fatty acid tails. So yeah, so just thinking about scale is important there. Okay, so if I asked you a question like that on the exam, you could probably logic it out, right? All right, indels, frame shifts. What is going on? What are these things? Why do we care about them? What the heck is happening? All right, so here's some codons in an mRNA strand, right? We've got our AUG, that's our start codon, woohoo! And then we've got all these other broies. So if we use that little crazy table to figure out what amino acids these codons form, we could figure out that it's this. Okay, this is our wild type, right? This is our no mutations. If I added these guys, if I inserted those, that would actually change the codons, right? So this codon didn't exist before. This codon didn't exist before. This is what we're talking about when we're talking about the frame shift. It's that triplicate of the codons is now being moved over, and so now we have all this crap that doesn't make sense compared to our wild type. Okay. Or no, oh, sorry. I'm getting ahead of myself. These are deletions. Ha! Okay, if I deleted those ones, then that's what would not make sense. Right? This is where we have that mistake. So these three are the samesies. This one is where we got wrong. So we just shrunk that up, effed up our codons. That's the deletion, the del part. If we had an insertion, same thing. We're having it shift, creating new codons. That's why indels are so detrimental compared to point mutations or some of the other ones, because this has these indels are more likely to change the protein that is formed. If we had a point mutation, if we change this A to a T, it might be okay, right? Because we have that redundancy in the genetic code. It might code for the same protein. So if that was a T, I don't know, I just pulled this out of my butt. I don't know if that's true, if it would still code for argon, whatever, arg. But, right, so we have that redundancy. So if we, point mutations are usually tend to be okay. These indels that cause a frame shift, those are what's detrimental. Okay. All right. A lot of people were like, oh my God, active transport, I'm gonna die. Here's what I want you to know for our class. Active transport requires energy input through the form of that energy molecule that we call ATP. We have active transport creates electrochemical gradients that power proton pumps so we can produce more ATP. We'll talk about the details of that when we talk about cellular respiration. So kick that can down the road. There's different, active transport happens in different flavors. We can have these integral proteins that are embedded in the plasma membrane. We can have this thing called exocytosis. So does anybody want to be brave and tell me what exocytosis means?
Excretion of waste.
Yeah, so we just like say there's crap in their cell that we don't want. We surround it with phospholipid bilayer stuff, and then we just like bloop it out outside of the cell. Endocytosis.
Cell eating.
Yeah, cell eating. It's the same process in reverse, right? There's something that outside the cell that we want, grab it, bring it in, goes wherever. Maybe we take it all the way out to the other side of the cell. Or maybe we take it to a lysosome. So that stupid virus that won't make us sick, and then we'll just get broken down and ha ha ha. Now we can use those organic molecules in our own bodies. Okay, endocytosis comes in a lot of different flavors. There's pinocytosis and phagocytosis and blah, blah, blah. You guys can tell me those, right? Or you can tell me them on Thursday. Okay. Okay, so don't get too bogged down in the details of all the stuff, you know, when you go on to take biochemistry and whatever. Then you can worry about the details, but for right now, we're learning high-level basic stuff. So that being said, here's a little video of these two are the Amoeba sisters, the little cartoons. Those videos are really good because they're cute and they get all the details right. And they're like entertaining. And in this crash course in biology, this one's really funny. This video, I highly recommend it. So there's the link for that for transcription and translation. That guy is hilarious. So if you're still struggling with those, check those out. There's a little bit more details than we need for our class, but at least it's an engaging way to learn those. So those are readily available on YouTube. Yeah. Okay, that's what I got so far. Now it's your turn. So we can pull out, we can do whatever you guys want. The rest of the class is yours. We got an hour. We can pull out cell models. You guys can ask me things. We can draw stuff out on the whiteboard. We can do whatever you guys need. So Zoomies, let me know if there's anything that you guys want me to cover or clarify or anything like that.
I
guess what I'm curious about is like, what are possible essay questions? Are gonna be?
Okay, okay. So I dropped some hints,
You did.
right?
No.
Does anybody remember what hints I dropped about possible essay questions?
One of them was, goodness, what paper was it?
Welcome to my life.
One question, why is the portion of the genome that codes for proteins that becomes ribosomes a good code for DNA primers? I feel like that was one.
That was one.
I had really interesting.
Mhm, mhm. So, you know, a good essay question would be kind of making this connection between the theory and the practice of transcription and translation. And then incorporating that, bringing it back in and grabbing ribosomes. Why do we care about ribosomes? Why are they important? Why do we have conserved regions? Why do we have variable regions? That's a good one. Let's see, I'm trying to remember what other hints I dropped. So, any things that we could like are incorporating and synthesizing multiple concepts. So, we talked about, let's see, let me think if I can think of one. Incorporating the structure of DNA and DNA replication. Right, that would be a good one. Talking about, there's certain organelles that have double tube membranes. And they also have their own DNA. What does that mean for the evolutionary history of prokaryotic and eukaryotic cells?
And that was that theory that it was its own thing at one point. And it got gobbled up.
Yep. So, and it's called symbiosis, but I don't think I've ever put that in any slide or written it down anywhere. But basically, yeah. So tell me, what are the two organelles that have double membranes, have their own DNA, have their own ribosomes? Mitochondria and chloroplasts. Okay. So mitochondria and chloroplasts, we think the evidence suggests, and there's a theory, so like a real scientific theory, not a hypothesis, that these organelles used to be prokaryotes that were free living. At some point they got gobbled up, and instead of being broken down, they were like, oh, well, I guess I'm your slave now, so don't kill me. And then that's how eukaryotic organisms evolved. It's because we like trap these suckers and make them slaves. So what does that tell you? So do plants have mitochondria? They have chloroplasts. Do they have mitochondria? They do. Yes. Plants do photosynthesis and they do cellular respiration. In order to do cellular respiration, you have to have mitochondria. They're the powerhouse of the cell. All right. So what does that tell us about when animals evolved and when plants evolved? Because do animals have mitochondria, right? Do animals have chloroplasts? No. So think about, well, you know, how that would have happened? Because we have these two endosymbiosis events, right? They get one to get mitochondria, one to get chloroplasts. But our animal line would have already branched off before that second event. That would be a good essay question. Let's see. Let me see if I can think of about a good essay question about plasma membranes. Talk to me about surface area to volume ratios. What the heck is that and why does it matter? Diffusion rates, all about diffusion, man, right? So what do cells want to do? Do they want to increase their surface area or decrease it? They want to increase it, right? Because we don't want to use active transport. We want to do it the lazy way, so we don't have to use energy. What are some of the ways that we talked about in class of cells maximizing and increasing surface area to volume ratio?
Microbillion.
Right, so we have all that folding that we can do of the plasma membrane. Perfect. What about size and shape?
They're small, so that way it doesn't take forever for food or molecules or whatever to enter. And get where it needs to go.
Exactly. That's why cells are small to have that large surface area to volume ratio, because the distance of diffusion is one of those factors. We want diffusion to happen quickly, so stay little, have lots of surface area. We talked about, we measured.
One little thing.
Yeah. So we had our cube, right? This is not a cube, this is but this is what I had handy. So this, we had our cube and we compared it to a cylinder. What did we find out? Even though they have about the same surface area, what did we find about the surface area to volume ratio in these different shapes? The roundy one had a higher surface area to volume ratio than the cube side. And so that's why we tend to see cells as spheres. Although plant cells are weird and they're boxy, but that's because they need to stack up, so they don't have skeletons and crap. Go ahead, Cody. What were you gonna say?
Oh, no.
Okay. All right. So that would be a good essay question.
You can have extra credit on this one?
I am. Yep. So the sources of extra credit are, so you'll have two options for essay questions. If you only have to answer one, but if you want to answer both, that's boom, 10 points. And then I do have, some other little questions that because I think I'm funny. So, so yeah, there will be 4 extra credit questions, and then maybe 5 if I'm feeling nice.
That's a good one.
Yeah, 5 is a good number. We'll make it. Well, yeah, we can round it out to a nice 15. Yeah, sure. Okay. This class is a democracy. We'll do that.
You go over cytoskeleton.
Oh, I can try. Okay. Cytoskeletons are also hard for me because I'm a plant guy and cytoskeletons, plants don't have cytoskeletons. Let's find out where I put that. Let's see, when was that? Structure and function of cells. Perfect. This one. Oh, I could ask you questions about osmosis. There we go. Okay, so here's a teacher hint. I'm not great at cytoskeletons, therefore. Okay, but you guys do need to know this. important for, especially if you are an animal people. So cytoskeletons really, plants don't kind of have them. Their structure comes from their cell walls, and so they don't have all of these components. But the cytoskeleton in animal cells is formed by these centrosomes. This is the major part of them, and it's particularly important during cell replication, which we'll talk about later. We'll talk about mitosis, meiosis. But this is kind of where everything connects to these centrosomes. And then let's see. Okay. I think there's a whole section about it, right? Yes, okay. Okay, let's see here. Okay. I have to lose a layer. Getting hot in here. Pardon me. Okay, so the thing that we want to know for this class is that there are three major components. There's these aren't going in order of size, which is annoying. There's microtubulars, and even though it says micro, it's the biggest. Okay. And then we have our intermediate filaments and then our microfilaments. And then this is what allows, stuff to move around. Okay. So our actin filaments, okay, blah, blah, blah. You guys can figure that out. That's their size. Okay, microtubules. These are our largest ones. These are composed, there's these hollow tubes, kind of like straws. They're composed of these particular tubulins, which are certain kinds of proteins. They have a positive end that allows so just like we have directionality in DNA synthesis, microtubulars also have directionality in how they grow. So we're always adding to the positive end and then taking away from the negative end. And so these are usually really dynamic. And as the cell is growing and changing and doing its cell stuff, these are growing and shrinking and growing, depending on how that needs to happen. So this dynamic instability is the way the word to describe that flux and the length of these things. And then this is a nice little YouTube video showing that in real time in an actual cell. So it's pretty good. Definitely, this is worth 30 seconds of your life for sure. In fact, let's watch it right now.
Writing's not that easy,
No.
but Grammarly can help. This sentence is grammatically correct.
I accidentally paused it. So you can see how these are being formed and changed super dramatically. And they're all kind of attached at some point to the centrosome. And it's just very dynamic. Okay. And then there's the credits, which we don't care about. Okay. So they determine, so we have all these different shapes that do all these different jobs. And it's these tubulars that tell us how that our cells are going to be shaped. It also tells us where our organelles are going to be. And then again, we'll talk more about these centrosomes when we talk about mitosis and meiosis. So keep them in your back pocket and pull them out again later. And then they also help the cells move around. So intermediate filaments, they have this nice little rope-like structure. They hold the tension that gives us our cell shape and our structure. These intermediate ones are unlike the other ones where they don't have that dynamic instability. Once they're formed, they kind of stay there. And then they're made of various kinds of proteins that do various kinds of stuff. The keratin in your skin is an example of that. So it gives it mechanical strength so that you don't, when I do this, I'm not smashing my cells into bits. And then it provides anchor points for those, the cell membrane and the nuclear membranes. So when we talk about the Golgi apparatus and whatnot, you know, it's hooking into the skeletal system. Again, our microfilaments, these are the teeny tiny ones. They're made up of actin, and then again, they have that dynamic instability where things are growing and shrinking from their growing in the positive end, shrinking from the negative end. And they also are attached to that centromere, the centrosome, sorry. Okay, same stuff basically, creating shell shape, strength, moving stuff around, and involved in cell division. And then you can talk about these motor proteins, which are cool, but you know, these aren't really benefits. Does that help, Cody?
Yeah.
Okay. Hey, anything else that you guys are feeling like you need a little bit of a nudge? Okay. So if I, whoops.
How does double helix structure relate to replication?
Good question.
There's too much information. What do you want to say?
Hey, who's the dudes that figured out the double helix business? Watson and Crick, and then who's that other person that doesn't get any credit? Okay. So we've got our nice little double helix structure, right? So tell me the components of DNA. What's on the outside here?
Phosphorus base pairs.
We got our phosphorus group. And then what is this little guy? That's our 5-carbon sugar, right? So that the phosphorus sugar backbone creates this. And then hanging out in the middle, we got what are those guys? We got our bases, right? And they use that complementary rule that we like, that we love. A's and T's, C's and G's, they hang out. Okay, so in Watson and Crick's paper, they're like, hey, now that we know it's like this, we know how it replicates. Cool, Watson and Crick, how do we do that? How does DNA replicate? Like which, so the choices are, we can have a conservative model of replication, we can have semi-conservative, or we can have distributive replication. What is it? Semi-conservative. Okay. Here's where my cute little zippers come in. So we can, unzip your jeans, right? Helicase those guys. And then, so here's our OG strand. And then now we can shift this enzyme into being polymerase, right? And so now it's incorporating the gray strand. So now we have that semi-conservative thing. So this would be our phospholipid, or nope, sorry, let me say the right words. This is our phosphate sugar backbone right here, right? And then the teeth of the zipper would be our bases. So because it has this double helix, it already has a template, right? Because that complementary rule, like we know what the next guy is gonna have to be because of that complement. And so one strand can be the instructions for building the second strand. If this was so one of the alternative ways that when they were in the 50s, when they're trying to figure this out, is that they had the sugar phosphate in the middle and the base was sticking out. Would we be able to have a semi-conservative replication using that way? Would that be so make sense? Not really, not as much as having this double helix with the bases pointing in. Okay, so basically, because we have a double helix, we automatically have a template to make a copy that's accurate.
And that's how they ruled out the triple helix.
The triple helix didn't match Rosalind Franklin's x-rays. And that's the one where they're like, hey, look at destruction. You know? So it was really Rosalind's data that cracked the case. And she didn't get any credit for it because, you know, she was a girl. Girls can't do things. James Watson has a book called The Double Helix. If you guys are interested in it, it walks through the discovery process of that. It's an interesting story, but you also realize what a big freak Watson is. And he's taken a lot of heat over the years for being sexist and all right, anything else you guys want to talk about? Yeah.
Sanger sequencing?
Sanger sequencing. Okay, Sanger sequencing. This is where we have DNTPs and DDNTPs. What's the difference between a DNTP and a DDNTP? Does anybody remember?
DD has to die.
Yeah, DD is di-deoxyribose, right? Go ahead, Cody.
And then the other one is yeah.
Deoxyribose, right? Deoxyribose, that's where DNA gets its first name from, right? That's what our sugar is. It doesn't have an oxygen atom on the 2' carbon. And then a di-deoxyribose doesn't have a oxygen on its 2 or its 3'. All right, now we got to talk about 3', 5'. First of all, how do we figure out if I drew here's our little 5' sugar. Five carbon sugar. How are we gonna figure out what carbons are which?
Start at 12 possible.
Yep. Here's our oxygen. Here's our hiding carbon. It's gonna be, we're gonna call it 1', 2', 3', 4'. I'm gonna move. Hey Zoomies, I'm gonna move you guys so you can actually see what the heck I'm doing. Let's go for a ride. Okay. Focus. Maybe. Didn't work. Cool. All right. So we've got our lovely little carbon here. We've got our things made. Deoxyribose. has no oxygen there. Dideoxyribose has no oxygen there. Why do we care that there's no oxygen on that 3' carbon?
Because it can't match up with the 5 prime.
It can't match your, right, because our next nucleotide, our next DNTP has to hook on to here. If there's no oxygen, there's no place to link it. So what Fred Sager did, dude, you are so cool, you get 2 Nobel Prizes. And so he figured out a way where we can incorporate, so we can have a pool of regular DNTPs. And then we can have these ddNTPs that are being incorporated at different rates randomly into, and so we're gonna have lots of different copies of the same DNA sequence. Some of them are gonna be really teeny tiny. Some of them are gonna be really long. Some of them are gonna be somewhere in the middle. And then because we know about diffusion, right? Molecular weight is one of those things that affects how fast you can diffuse. Because if you're a little guy, you can just zip on through easy. But if you're a big guy, you're gonna bump into stuff. And it's gonna take you longer to get there. So we can use these different size fragments of our DNA. And the other cool thing about the ddNTPs is that there's a little modification to each of our letters, each of our bases, that when you zap it with a laser, it lights up with a different color. And so we can just zap it. The small fragments get there first. And so we can say, okay, that's an A. And then the next fragment lights up with a different color. And so we can say that's a T. And then the next fragment gets there. And so we can use that, different rates of diffusion to, and these ddNTPs that created different sizes of DNA to actually determine the sequence of DNA. Freddie gets a second Nobel Prize. That, Camille, I think that was you. Does that help? Okay. We can pull down the cell models if you guys want to go over organelles. Whatever you guys want. It's your class right now.
There's something about osmosis, about the efficacy of it. Like, it kind of become, I just wanna-
Okay. Yeah, okay, so this is a good, yeah, this is a good one. Does not have to be- Oh, see, this is the right stuff. I don't know, we'll find out. Okay. So while I'm getting this ready, somebody throw at me the definition of osmosis. We're talking about water.
Moving the free water and also through semi-permeable membrane.
We got a semi-permeable membrane, right? So if we're not, if I'm asking you a question about sugar, the diffusion of sugar, you're gonna be like, well, that ain't osmosis. right? Okay. So kind of our criteria are water only. We have to have a semi-permeable membrane. Okay. So. And the, let's see, how can I say this? Let's draw some pictures first. I got a jar of stuff. Here's my semi-permeable membrane so that we can fit our rules. In this juice, I've got some sugar dissolved. Here's where it gets tricky. Of sugar, is this a high concentration or a low concentration compared to this side? This is high sugar. What does it mean about the water? The concentration of water in that? The concentration of the water is low on this side. So we could do the opposite here, right? So this would be a lower concentration of sugar. And a higher concentration of water. These little square brackets are the way that lazy chemists write concentration. Okay. Where are the sugar molecules what are they gonna do? Sugar molecules are going to try to diffuse to equal out, right? They want to reach equilibrium. The water molecules that are invisible because I didn't draw them. Let's draw some. What are they gonna do? They're going to move from high concentration to low concentration. Same as anything else. Just to make this confusing, that's when we bring in tonicity. When we're talking about osmosis, but now, oh my God, why? We're talking about the concentration of the solutes. Right? So does this side have high tonicity or low tonicity? High. And then this side would have low tonicity. All right, let's say I have a beaker of whatever. And then I have this little, I don't know, let's say a bag of dialysis tubing that I put in some starch, some, let's say glucose, and some salt. And out here, this is distilled water. So there's just water molecules in here. What's going to happen? What's starch going to do? It's gonna stay-
Pass through the semi-permeable membrane, but
It's
the water's going to replace the salt of sugar.
Yeah, so the starch is too big. So it has to stay. Glucose is brittle, right? So it's going to go from high concentration to low concentration. So we're going to have glucose moving out of the cell. What about salt? It's really little. Not glucose is little, salt's really little. So it's going to move out of the cell also. And then Cody, what's gonna happen with the water?
It's gonna move inside of the cell.
Yeah.
There's a lower concentration.
Right. So the water is going from high concentration on the outside, moving into the cell toward low. Okay,
So the one on the right is hypotonic, and the one on the left is hyper.
okay, so yeah, let's bring those words in. Okay, so this guy, let's say, what kind of tonicity does he have?
Hypo.
Hypo. Right, because the concentration of the solutes is lower outside of the cell. Okay. If we had a cell in here, this would be hyper tonic, right? Because we have all the, so if we forget about this side. Because we have more sugar outside of the cell, we have more solutes outside of the cell than we do inside of the cell. So that's a hypertonic. And so what would happen to this cell in a hypertonic solution? It would shrivel up, right? Because its water is going to leave. This is why don't drink salt water, guys. Don't. That's why it makes you more dehydrated, right? Because you're making a hypertonic. The water's going from high concentration inside your cell to low concentration outside. If we wanted a nice little happy cell, what would we want? What would we need? We need isotonic. Iso means samesies. So the total concentration of solutes inside the cell is equal to outside the cell. It's so we make these rules about, but we're talking only about water with osmosis, but then we immediately start talking about tonicity, which is the concentration of solutes. So then osmolarity is kind of like, you gotta like think about it in backward slam. So that's one of the things that where a lot of students will trip up. So remember, tonicity is the concentration of solutes. But when we're talking about osmosis, we're talking about the concentration of water. And those two things are definitely related, right? They're inversely related. Which just is like, one's on the numerator, one's on the denominator. When one changes, the other one changes in the other direction. So if you're confused, draw stuff out in the margins of your exam. Pull out some scratch paper, logic it out. Draw it out to increase your confidence. Don't just be like, yeah, so like that is what saved my butt so many times in undergrad, was being able just to like, okay, wait, hold on a second, I'm confused. Let me draw a picture for myself. Okay, now I got it. And for me, that's why drawing stuff out on the whiteboards is really useful, because that's how I think. So knowing how you think is key to being effective at studying. Okay, how are you guys feeling? I'm getting some trepidation waves, but are you guys more confident than the first exam? Less confident?
More confident.
More confident? Yeah, okay.
You're not overconfident.
So, okay. So I studied quantum-associated fungi, right? There's this hypothesis that's called the intermediate stress hypothesis. So if we look at a graph, we can say, okay, this is like a low stress environment, high stress environment, And then on the y-axis, we have the percent of colonization from these plant-associated fungi. So in a low stress environment, you can't afford to pay your symbionts, so you don't have very many of them. No, sorry, let me say that the right way. In a low stress environment, you don't need symbionts because you're fine, you're happy, everything's cool. You don't need those guys. In a high stress environment, you can't afford to pay your symbionts, so you don't have them. And as you increase in stress, you're like, oh man, I gotta start hiring some help. Until you get to this intermediate. But then once it starts to get more and more stressful, then you can't really afford to pay help anymore. Like, so why would you pay somebody to do something you could do for yourself? Why would you pay somebody when you can't afford it? But if you kind of hit that middle part, that's when you have the most symbionts. And the whole reason that I tell you this story is to say the intermediate stress hypothesis works for taking exams too. If you're too chill, this is what I found for me. If I went on days when I was like, fine. I didn't study enough and I didn't do very good. If I was too stressed out, I would overthink things and I wouldn't do well. But if you kind of hit that sweet spot where like, you know, you got to study, you got to put the work in, but you're not like freaking out, that's when you do the best. So work toward that intermediate stress. At some point you have to be like, okay, it is what it is. But you know, you don't want to be too chilly about it either. All right, so we still got 20 minutes. If you guys want to leave, you can. If you want to hang around still, you can. I'm here for you. Whatever you need to do. We'll have Tracy and Katie help you guys make up the quiz before you so don't bounce until we do that. But the rest of you, if you guys have more questions, I'm happy to answer them. You want to take some time to look, we can pull stuff down. Whatever you guys want. How are you guys doing on Zoom? You guys have any questions for me? Have anything come up while we were doing those discussions?
I'm good.
Okay, great. Carson, do you have anything? Okay, can I see yours? All right, so happy studying, you guys. I'll see you on Thursday. So the exam is the only thing that we're going to be doing on Thursday. So once you get that done, you can go and have a nice spring break. Great, thanks guys. See you Thursday.